Firebase, I don't know what it is. But yeah, so going back to at least maybe not to the primary data, but at least to a paper that describes the primary data. Yeah, and then you can track back where the logic comes from of deciding that there should be an arrow here, there should not be another arrow here, and another one over there, and they mean something. Yeah? Okay. Any questions about this? Okay. I'm not sure if that's a good thing. Right. Oh yes. And it's also, if now we're talking about these symbols, it's very important that you have a vocabulary that describes what all these elements mean. What do the rectangles mean? What do the arrows mean? Do all the arrows mean the same thing, or does it depend on what type of entities they connect? You have to think about what that means. It's easy to draw a diagram of the citric acid cycle, but to be sure what all the symbols mean that you've drawn, that's very important. It's certainly important if you're going to use this in a machine-readable way, because then there's no human who can say, oh, wait a minute, this is an enzyme, but that's actually gene regulation. Yeah? Both could be represented by an arrow. They mean something very different. And if your analysis method takes this data as meaning the same thing, you might draw a very, very strange conclusion. Yeah? Okay. So these are all things that if you want your data to be reused. So why is data reuse important actually?
Rerun experiments.
Sorry, to
rerun experiments.
To rerun experiments, but that means you're actually generating new data.
Or maybe someone wants to check if.
Oh, you mean like a computational experiment, like a analysis pipeline?
Yeah,
So you want to be able to run the same analysis again?
Yeah.
Yes. But that sounds like it's about going from the primary to the derived data. So if you want to be able to go back to the primary data again and rerun, or maybe make a variation on the analysis to see, or a better analysis tool and run the same kind of analysis but with a better tool, see if it's actually better. Yeah, there's much more to data reuse, because when you run an analysis tool, what do you get out? A hit from, well, yeah, but more generally, you get more data out. And that might, at some, you might actually, it could be that you say, okay, I predict that this gene has that function. Well, if you do it for one gene, it's not a lot of data, but you could do it for a database of genes, and then suddenly you have a database of predicted function, predicted gene functions. Yeah? That's a very simple example, but there are a lot of these things. You want this data to also be reused by other people that are looking for gene functions because they want to interpret their tumor data or something. So the data reuse goes much further than just be able to go back to your primary data and run a better analysis or something else. It actually goes about all our data. And so a lot of the work at this moment is about making that possible. So what typically happens, and I've spoken to at least 5 people in the last 2 weeks that stumbled on this issue in their own lab, that when a new PhD student comes in to continue on a project, they'll typically have to throw away some of the data because they don't know any longer what it means. Half of the scripts because they can't read them, or if they try to change anything, everything breaks. And then write a lot more scripts because the next data set is slightly different. It breaks everything in the old scripts. So months and months and months of researcher time are being spent in reusing scripts and data, programs, tools and data, even within one lab, where you can actually go back, you can probably still, there's a lot of people who know what the previous PhD student did. You can probably still get his email address or her email address and email them. Can you tell me what this big Excel file was about? Because it doesn't have column headings, it just has numbers. You can laugh about this. This happens. This is not an exception. There's hardly any project where this doesn't happen. It's getting better slowly, but a lot of effort is going into this. It shouldn't have to be this way, right? Because this is not a very, very useful way to spend your time. Okay. Okay, so let's see, where are we going? We're almost going for a minute. So so that's that side. And the good side is that there's increasing effort, and this is going on for decades already. Growing community of institutes and people that are trying to make solid, reusable data resources. And these are three of the main players. So the EBI, European Bioinformatics Institute, which is part of the EMBL, European Molecular Biology Lab. I think it was founded somewhere halfway last century. The EBI is founded a bit later. So they have a lot of research. Who's not heard about the EMBL before? This is Europe. In the US, they have a similar thing called the NCBI. I don't remember what it stands for. Anybody knows?
National Center for Biotechnology Information.
National Center for Biotechnology and Information. Yes. It sounds sort of familiar. So what we call bioinformatics in Europe, they call computational biology in the US for weird historical reasons. And so they have lots of resources. If you've run BLAST, you've run it either probably via the NCBI website or via the EBI website. They both host BLAST servers. They both host the search databases and a lot of other resources. Then this is a journal, the Nucleic Acids Research Journal, and they have an annual, once every year, they have a database issue with new papers about new databases or describing updates of existing databases and so on. So these are the three places where, if you're looking for a particular data resource, you should start. PubMed is NCBI. Who has not used PubMed? Sorry, it sounds slightly accusing in a way, but it wasn't. So this is your search engine, right? User is a database. Yeah? You can think of it as a library. The library is just a database. If it's not indexed, it's still a database. It's just bad design one. A taxonomy browser. Just some examples. This is EBI. It looks like they have more, but this is just because it's Europe that we show more of that. The 1000 Genomes Project Database, European Nucleotide Archive, ENA. Sounds weird if you say ENA. It's like you're missing one of the two letters. Ensemble, so that's genome annotation. The EGA, European Genome Phenome Archive, so that's a lot of genetic data. PFAM families, we're starting to look into that. PROT, that's the old, it used to be called SwissProt, that's the oldest sequence database in the world, that's still alive and kicking. Oh, speaking about alive and kicking, and then we can go for coffee. One of the big challenges in bioinformatics, I would say actually in biology as well, because you can't really do biology without all these resources, is this is considered by the funders, funding agencies, infrastructure, and it's very hard to get funding for infrastructure. And more so because what the funding agencies classically consider as infrastructure is high-speed internet connections, compute clusters, but also experimental facilities like genome sequencers, high-end microscopes, these kind of things, NMR machines. That's infrastructure, right? Now, our infrastructure, bioinformatics people, is these databases. But the cost of these databases is not in the hardware. Well, a bit. The cost is in the curation. So our infrastructure, data infrastructure, is people. And then you go to the funding agency and say, well, we have this incredibly important. Nobody can do biology research without the network that be up and running. Okay? It just doesn't work because all the sequence annotations are there. And then you say, well, we have a team, we need a team of 100 full-time annotators to maintain this resource. This is infrastructure, because they're not researchers in the sense that they produce papers. They consume papers and turn it into a database. And then the funding agency said, yeah, but the people are not infrastructure. Well, there's a nice thing to say about people, but it's a terrible thing to try to manage the database. Okay. This is not something that you can solve now. It's also not something that a lot of people are trying to solve this. Maybe one of you in the future will remember what I said and can help us solve. Okay. Nucleic acids database research, I already said it as a journal. There's some links to find entries. There's lots and lots and lots more databases, and then there's even more. Yeah? I think that's enough. Next, I'll go into, so we've had this column now, we go into the scope, but before that, I will have coffee. Yeah. Unless there are urgent questions right now. No? Okay.
Not more urgent than coffee. Not more urgent than coffee. Kon naast zitten, ja? The others, I guess.
I know what it is. I just
There is a side. It's full here. There is. I don't.
It looks like it's still.
I can do some strange face. On YouTube, my face. No. Anonymous.
你去看了那个。
I think it's like €1 or something.
Maybe sit here because you're not so tall. Otherwise, I'm sitting in front of the camera. Sorry, but you could sit here, I guess. You're not mental.
Sit here. Maybe you can sit here.
Okay.
I don't know. I think this is Isabella's sense.
Isabella 来了。
这个有人，这个没人。
好的。
这个有这个。
你看，它能被抬起来吗？不能。
你可以试试看，你可以把那个把那个小心要掉下来。
Oh no.
今天怎么没有看到那果汁？
哦，那是午饭呀。
午饭啊，那是午饭啊。
这个就有2杯啊，
拿保温杯
这
装的。
那个是
热的是什么呀？
咖啡啊。
哦。
不是说的椰油咖啡吗？
你要减肥吗？
对呀、因为最近不运动啊，这个今天是橙汁，
就这样比较直接。超市
对啊，
里自己买
很便宜。这个就橙汁一大袋，可能10个才几块钱吧，2块多。
可以榨多少？但是我总觉得
一个这样一杯。
哦，一个，那你是果汁果肉融合在一起。
因为你吃的时候本来也这样，把皮剥掉，
对，
全部都吃了嘛。
那也可以直接吃，直接带个橙子来吃。今天讲了什么？
讲那个数据库，就是那3个数据库，但是他还没讲到呢。
哦，还没讲到那里，讲够了吗？
他就是讲，为什么，就是说，就是做研究就是生物什么什么各种东西的话，他先举了个例子，然后是需要哪些数据什么之类的。
It's okay. I'm gonna stand for you.
But you were going anyway, right?
Yeah, I'm gonna sit there.
Okay.
Me again. Okay. might as well do.
I think I can pass. Thank you.然后我前两天
It's more convenient you can sit here. Yeah. If I sit here, then I'm completely in front of the camera. But I guess. I guess you can put your laptop here. Oh, yes, sorry.
sorry.
Oh, ja.
Ik weet wel je moet doen.
Alleen dat vind ik irritant, dat je niet zo bent. Maar je kan het gewoon bij Apple, het is gewoon command, oké, dan kan je gewoon binnen.
他让我坐下面，他觉得
bij Windows 7。
这你可以看到
对，我不太敢讲话上课的时候，因为我觉得英语太差了，或者讲的东西错了，然后他这个录进去，然后上传。
行，那你10年之后，假如说还有这个video，你还能看到自己当年多好啊！把我的包给我。
这个吗？这个。
Oh, sorry. It was like in the morning, you would smell the like, oh, I don't have it. Now I'm thinking about they do it. It's really good.
昨天我回家就基本上啥事儿没干。
你不是在编程吗？
我是昨天下午没走进去看，但是我并没有出来什么太多的看。
Is there a I'm gonna strike and then so this is my brother's first. Maybe people don't know what's up.
然后我回家就彻底放纵。
我在想，因为我们组那个 Sarah， 她昨天不是我们都没开始上然后我就在那里看生物的东西，然后我打算回去的时候，他就说他已经把就是今天的那个。
Yeah. Alright. This is your last one.
David。
我的衣服还在那个里面。
Taking a picture.
Oh.
Okay, success.
how many
Right. Okay, welcome back everybody. Geontology, go. What do you know about go? It's a question to you. It's about genes, it's ontology. What else?
Oh, I'm sorry, I didn't hear the question, but I get it now.
What do you know about go? Information about species? Not quite, but it's a bit information about genes and their function. Yes. I don't think there's anything about species in go.
Can't you find more where they are in other species?
Yes, you can. So you can find, you can use go to trace genes of similar function between species.
Yeah,
Yes. Okay, but that's what you can use go for. That's also nice to know. But what is in go? What is? How does it work, or how is it built up? So processes associated with genes. Right? Oh, just by the way, there was a question here from the front row. There's also just the plain English word homology. If you look it up in a dictionary, that of course has a relation with similarity. Yeah? But this is the molecular biology term of homology. Right? Don't confuse those two. It's very annoying that they have a similar appearance. Yeah, but they don't have a common, well they do have some commonalities. But that'll make it more confusing. Okay, go. Processes. Processes. So it's about genes, right? What else? Sequences. Not strictly. It has to do with sequences. We'll come back to those in a minute. What else? Functions. Yeah? What else?
Location.
Locations. Yes. So these are the three main, I don't know the word for it, the three main things in Go. So location is sort of obvious, right? Is it in the nucleus or on the nuclear membrane, envelope membrane, or in the plasmatic reticulum, or in the Golgi, or in the cytoplasm, or in mitochondria, or mitochondrial membrane, or on the cellular membrane, or extracellular, or a combination of those. Yeah? Some proteins are in different places. What's the difference between process and function? That's a hard question.
Function is the exact function of the sequence you're looking at, and process is what is involved with, amongst other proteins, functioning.
Yeah. I'll have to repeat what you said because I don't think anybody in the back heard it. This is just not possible from that. There's nothing wrong with it. So the function. Not sure about that. Okay, I don't have any evidence to conclude that there's anything wrong. That's very weird if you say. It sounded all right. Okay, so the function is about let's say what the molecule does, right? So it's associated with this gene and the direct function it performs, right? So a gene can be, for example, a transcription factor, or it can be an enzyme doing alcohol dehydrogenase, which is my favorite example. Or it could be a extracellular sensor protein. Yeah. So that's the function. So this is directly related to what the molecules do, or what the molecules of that type do. Then the process is what they're involved in. Yeah? So you could say, what do they do it for? Yeah, so what does this transcription factor do when it binds to its target gene? Or what does this membrane sensor molecule sense, and what does it relay the sensation to? Yeah? So it could be an opsin in your retina relaying the reception of a photon to a signaling cascade that translates the signal into a neural pulse. Yeah? I don't know the biological word for that process, but that's the process. The sensor, membrane sensor molecule is involved, but there's a lot of other molecules involved in the same process. Yeah? Okay. Where are we? Yes. So there's a whole consortium behind this to actually to maintain the Go database, because it's not a static thing. There's new things being added and modified. So why do we need Go? Oh, yeah.
Is the location about where the gene is located in the genome, or
No.
where the gene is expressed?
No, it's not. Well, the gene is always expressed in the nucleus. It's not about in which tissue the genes are expressed, because there's genes that are only active in the brain. There's some genes that are active in the brain and in the testes, which in man there's genes. It's true. I can probably name any pair of tissues, and it's true. There's some genes that are only expressed in those two. But that's not about. It's about the cellular location. So it's where the protein. No, it's where the gene product is. So the Go defines things in terms of genes and gene products. Gene product typically is a protein, but there's another possible gene product. What's the other one?
RNA?
tRNAs, for example.
Yes, tRNAs or small interfering RNAs or lots of, so it can be protein or an RNA. If you make protein, there's always messenger RNA involved, but that's not what you mean by the gene product. Yeah? So it's any RNA except messenger RNA or the protein, more or less. Okay, so and so it's where the gene product performs its function. Yeah? Because it's also always expressed on the ribosomes, which in eukaryotic cells are on the endoplasmic reticulum. So it's also not where they're expressed, it's where they go to do their function. Okay. Yes, so why do we need this? And that's actually, who said it? I think that's what you said. About species, you said it. Thanks. Because there's an enzyme in Drosophila, fruit fly, that's called geranyl transferase. Geranyl is a small molecule. I think it's a pheromone-ish like molecule. Somebody probably knows better than me. And it does something to this molecule. Yeah, so it's an enzyme. In humans, we actually have the same enzyme that does the same function, but it was labeled with a different name because different people there's different people researching this protein in Drosophila than there were people, and different people researching this protein in humans. So they got a very different name. It basically does the same. Yeah. So how do you find out? Yeah. Well, this is one of the things. Yeah. But then, so if you actually get these people to talk together, they might find out, hey, these two proteins are actually doing the same. Yeah. But now this is tedious. So if you want to be able to do this in a more global way, preferably in an automated way, you would like to have a standardized annotation form that allows you to compare molecular function between different genes from different species. Because now you have the, so this was the original text annotation of these two genes. But now if you start annotating them with the gene ontology database terms, then you should find out that this one is annotated with almost exactly the same terms as this one. Thereby you can conclude that they have the same function. Yeah? And then certainly if you see that they're also involved in the same kind of processes in both species and in the same location, then you can say, oh, these two genes are actually doing the same. Yeah.
Then you still do not know if they're chemically the same thing.
The proteins.
Yeah.
Yeah, so how would you find out whether the proteins are similar?
Yeah.
You can compare the structure if you're lucky. So a lot of these enzymes have been studied for ages. Well, maybe not for ages, for decades. And somewhere along this route also people solved the crystal structure. But you can do something simpler.
Look at active site sequences.
Active site sequences, or just at the whole gene sequence. Right? If the sequences are similar, then the proteins are similar. Right? So now, when can you conclude that these two proteins are homologous? Meaning that some common ancestor of human and Drosophila actually had a gene that was doing the same function, and it evolved to be this gene in Drosophila, and it evolved to be this gene in human. When can you conclude that this must be true? Based on the Go annotations? Then you know they have the same function, they do it in the same place, and they do it for the same reason. Yeah? Just those three. Can you see my pointer? If I, yeah, I can't see it from here because the light is reflecting. Okay. So I don't know where I'm roughly. Am I roughly in the right place?
No, on the right side of the board.
All right. So now this is better.
No, it's in the middle.
Well, you get it, right?
Yeah.
I'm pointing to. Okay. Sanne warned me, there's a lot of these databases things and enumerations that don't make it too boring. So I'm trying to switch in some things to away. Okay, so where was I going? Oh yes, so you know from Go that these proteins are doing exactly the same thing, yeah? As far as, I mean, for a reasonable definition of exactly. So when do you know that they are homologous? You look at the sequence, right? So now the two sequences are completely different. What can you conclude?
They might still be homologous.
They might still be homologous.
Have changed a lot, but like if you find similar sequences, then you know, okay, yeah, probably homologous. But if this human and
Yeah.
guys are quite far away.
Just a second, because otherwise I have too much to repeat. Yeah, because you're getting it right. So if let's say these sequences are similar, yeah. So would it be likely that they are not homologous? They're doing the same thing, and the sequences are similar. Yeah, maybe 60% similar. So 60% of the residues are the same. Could you get that situation without a common ancestor?
Guess that's
You
possible.
could, yes. Is it likely? No. Why not? If you have a protein of 100 amino acids, how many different types of amino acid are there? 20. So how many different sequences can you make of 100 amino acids? 20 to the power 100. Yeah? That's 2 to the power 100 times 10 to the power 100. 2 to the power 100 is about 1 million. No.
No, it's a lot more.
Yes. It is 2 to the power 10 is 1024.
Yeah.
2 to the power 100 is. Is that right? Oh no, it's
It's a lot more.
It's a lot. Yeah, you get the point. Yeah, even don't forget about the 2 to the power 100. 10 to the power 100 is already a lot. Yeah.
It actually happens quite a lot where viruses or bacteria take on genes, like with horizontal gene transfer, and then they optimize them for themselves, so they have a completely different or mostly different sequence, but it's still, the origin is the same.
Yes. So you can have the same origin and then diverge to the point where from the sequence you can't recognize that they had the same origin. Yes, that's possible. If they still have the same function, that it gives you a clue, right? Then so now you look. So if the sequences are similar, it's easy. It's very unlikely that they're not homologs, right? So that's also why the confusion between common ancestry, which is you can still read it at the bottom, and similarity as the definition of homology comes from, right? Because if two things are similar, it's very unlikely that they're not homologous. But that's not the definition of homology. Yeah? Okay. Now consider the other situation where the code annotations are nearly identical, but the sequences aren't. They're very different. Yeah. So that means if you align them, about 1 in 20 residues, 5% are the same, which is what you expect randomly, because if you pick 1 out of 20, then you have 1 out of 20 chance that it happens to be the same as the one in the other protein. So they're indistinguishable from random if you compare them. Is there still something you can do to get an idea of whether they might be homologous but diverged a lot, like for example through viruses with horizontal gene transfer, but it could also be to very distantly related species. Which data could you use to say something about that? Which types of data are we looking at now that I described already?
Sequences.
We have sequences here, because we compare them. What's the other type of data we have here?
Structure?
Yeah.
We haven't, yes, that's the other one that we don't have yet, right? The one that we do have over here is what? Function. Yeah, so they have the same function, they don't have the same sequence. Now you look at the structure.
FASTA?
Well, the FASTA is
You
sequence.
can also look at the amino acids.
Yeah, but that's the sequence.
Well, that can be different than the DNA sequence.
No, okay,
DNA
but
sequence is different than the protein.
when we're looking at, when we're talking about the function of a protein, it doesn't make sense to look at the DNA sequence. You want to look at the amino acid sequence for the reason that you were about to say. You can look at the structure, right? Now suppose the structures are quite different. What is your conclusion? Functions are the same, structures are different, sequences are different. It could still be homologous, but is it likely?
No, that's nothing.
Because it's even stronger for structure. For two, sorry. What am I going to say? No, yeah. Oh, sorry. Yes. If you have to do it the other way around. So if you start with a common ancestor with a certain sequence, certain structure, certain function, it starts diverging. There's actually really only one way to keep the function. You can change the sequence as much as you like, but if you start changing the structure too much, it's very likely that you lose the function. Yeah? Get it? Okay.
What is the active site in the sequence?
Yeah, so the working horse of an enzyme is the active site, right? That's where the ligand binds the geranyl or the farnesyl molecule where it binds. That's the active site. That's where the chemistry happens in an enzyme. So you could have the situation where almost everything in the structure changes, but the active site is the same. That is very hard to do for a protein. Yeah? So that doesn't happen at all. It could happen so that also first the sequences diverge, then the structures diverge, function might still be conserved. Yeah? That doesn't happen a lot, but it can happen. Okay? So now so that means if the structures are different, they're not homologous. Yeah? Well, most likely. If the structures are similar, no matter how different the sequences are, if they serve the same function, then they're most likely homologous. Then there's some very interesting exceptions to that, but we'll go into that in the structure bioinformatics course later this year. Yeah, you had a question?
Yeah, is this then an example of convergent evolution?
Yes, so if you have two proteins that have the same function and they're not homologous, then it's convergent evolution. That means you started from two different starting points, and because a certain function can be very important for survival of the species, two different species in this case, they might end up inventing the same function with a very different molecule. Yeah? And there's plenty of examples for that. The easiest example to remember, because it's not molecular, is the eye. So we have our eye, the mammalian eye. The fishes have the same eye, and you can recognize it by the fact that it's built the wrong way around. Yeah, so if you believe in intelligent design, there's very many examples of biology with extremely unintelligent design, because all the infrastructure of your retina is in front of the sensitive layer. So the nerves and the blood vessels are at the front of your retina, and the light sensitive cells are at the back. If you design a camera, you would never do that. Right? You don't put the cables and all the stuff in front of the light sensitive chip, because it distorts your image. It makes everything very complicated. Squid, octopuses, I'm not sure both of them have them. I'm not a zoologist.